Hi, this is Mr. Worski, and today we start off talking about protein synthesis in our first screencast in the series. I know you're excited, so am I. If you have any questions, you can refer to the video again, look at your textbook, or maybe even ask me in class. So the organelles that we are going to cover, well, that we have covered, we've covered the cell wall, cell membrane, chloroplast, mitochondria, nucleus. Now we're going to cover the nucleus, the nucleolus, the ribosome, the endoplasmic reticulum, and the Golgi complex. Pretty much everything else in the cell we are going to talk about. I know, it sounds so cool. So protein synthesis is the production of proteins because that's what every cell does. Every cell, as you know, is a protein factory, and it produces specific proteins necessary for the functioning of the cell, as well as the tissue, the organ, and you. The gene is a segment of DNA that codes for a polypeptide. We're going to say protein now. That's not altogether correct, but we're going to say either polypeptide or protein and use it interchangeably. I know it's really bad, but we'll get over it. So the code on the DNA must be carried to the ribosome because we know that the ribosome produces protein because we talked about that when we talked about the cell organelle functions. But how is it carried? The answer is RNA. Here's an animal cell. Those are the rest of the organelles that we're going to talk about. This should be familiar to you. Love it. It happens in plant cells too, but I didn't want to bore you with plant cell pictures. So RNA is ribonucleic acid, and the monomer of a ribonucleic acid is a nucleotide, and the abbreviation is NTP. Remember, DNTP was the abbreviation for deoxynucleotide. The nucleotide is just NTP. So in DNA, we had deoxyribose here, but in RNA, we are going to use the ribose sugar right here. And we know it's ribose because it has that extra oxygen that arrow is pointing on right there. We know that RNA is also single-stranded. Because of that extra oxygen, it cannot form a double-stranded molecule. And it has uracil and not thymine. Right? So we told you about uracil and thymine. The difference is the thymine has a CH3 group. This uracil has a hydrogen only. That is the only difference between uracil and thymine, that little thing right there. So there are different types of RNA. Now, I know in 9th grade or 8th, 10th grade, we told you that there were only 3. We said there was messenger RNA that carried the code from the DNA to the ribosome. We said that there's ribosomal RNA that makes up a ribosome, and tRNA or transfer RNA, which carries the amino acid to the ribosome. And that's true. But there are different types of RNA in addition to the 3 that we talked about before. There's hnRNA, which is heterogeneous nuclear RNA, and that is pre-edited, pre-spliced, pre-modified mRNA. There's snRNA, which are small nuclear ribonucleoproteins, and those make up spliceosomes. They have a really cool nickname called SNRPs. Yes, I said SNRPs. S-N-R-P-S. SNRPs. miRNA is microRNA, which are small RNA sections that can bind to either RNA or DNA. And the same thing with siRNA, which is small interfering RNA molecules which can bind to DNA or mRNA. Now, miRNA and siRNA are ways that we regulate the proteins that we produce. And we will talk about them some other time in greater detail. I know everything is being kicked down the road, and I apologize, but you have to know the background before you can actually apply this other stuff. So the first part of protein synthesis is known as transcription, and transcription is we transfer the code from DNA to RNA. Now, the enzyme that we're going to use for this is RNA polymerase. That sounds like DNA polymerase because it is very similar to DNA polymerase. Now, in prokaryotes, there is only one type of RNA polymerase. That's easy. In eukaryotes, there are 3. The one that transcribes genes is RNA polymerase 2. RNA polymerase will unwind DNA, and as the DNA is unwound, hydrogen bonds separate between the bases, and the RNA polymerase will also match up free-floating nucleotides, NTPs, with the exposed DNA bases. Now, A binds to T, U binds with A, and C binds with G. Now, RNA polymerase does the job of two enzymes. Don't need helicase to unwind anymore because RNA polymerase will do that. And just like DNA polymerase, RNA polymerase will match up the free-floating nucleotides with the exposed DNA bases. Now, RNA synthesis or transcription happens in a 5' to 3' direction because remember, you can only add to the 3' hydroxyl group on the ribose sugar, just like DNA. It happens at a rate of 60 bases per second, and ligase will seal up the newly synthesized RNA strand. That was a lot. I know, but you can handle it. So here is transcription, which RNA polymerase is right here. It unwinds, puts down, and it happens in a 5' to 3' direction, which means that this is the 3' to 5' direction up here, right? G binds with C, A binds with T, U binds with A. So where does RNA polymerase know where to land on the DNA strand? Great question. These are known as promoters. Promoters are DNA sequences that allow transcription factors, that's what TF stands for, transcription factors, to land. The transcription factors act as a flag for RNA polymerase. So the transcription factors bind to the DNA sequences, and then the RNA polymerase will bind next to the transcription factor. Now, within the promoter sequence, there is an initiation sequence. That's where transcription actually begins. Now, before some initiation sequences are boxes known as TATA boxes that have a high concentration of thymine and adenine. The terminator sequence, where RNA polymerase will stop, is the TTA-TTT sequence. So to get out of this, just remember, places on the DNA are known as promoters. Promoters are where transcription factors land, and RNA polymerase will look for, if they could look for anything, transcription factors. That's where we'll start transcription. Now, transcription starts at the initiation sequence. Before that, you have the TATA boxes. RNA polymerase will stop at the TTA-TTT DNA sequence. So here is a picture talking about that. We're talking about enhancers, but we'll talk about that later. Here's the promoter, and the promoter, we have the transcription factor here. RNA polymerase looks for this big thing, binds, boom, and produces an RNA molecule. Now, in progs, the mRNA is made directly from the DNA. Okay, that's great. We learned about that. No problem. The RNA that comes off is mRNA. In eucs, though, in eukaryotes, hnRNA is made directly from the DNA. And hnRNA, one of those RNAs that are new, is pre-edited and pre-modified mRNA. Now we need to change hnRNA to mRNA because ribosomes use mRNA only. So you might be asking yourself, well, what has to happen to the hnRNA in order to be made into mRNA? And that is an outstanding question, and I'm really glad you asked. The hnRNA needs to be processed. Okay, so first thing you have to modify the ends. On the 5' end, there's going to be a special base called methylguanine, the modified guanine that's added to the 5' end of mRNA. On the 3' end, we're going to add a poly-A tail. Poly-A means many adenines bonded together. It's about 150 to 200 adenines that will be added to the end of the hnRNA to make it mRNA. And that helps protect from enzymes in the cell. Now, the special base at the beginning might help the ribosome find the beginning of the mRNA. The next thing that has to happen are introns must be spliced or cut out. And you're thinking, what the heck is an intron? Well, let me tell you. The average hnRNA molecule is about 8,000 bases. The average protein is about 400 bases. There are 3 bases per amino acid. So we're talking about the average protein is about 1,200 bases long. So most of the hnRNA doesn't even code for a protein. So this part, there are parts of the hnRNA that does not code for any proteins, and those are called introns. Right? So we have also parts of the hnRNA that code for proteins or code for amino acids, and those are called exons. So what you need to do, or the cell needs to cut out the introns and splice together the exons. How are the exons spliced and joined together? Well, spliceosomes. Spliceosomes are made up of snRNA, or SNRPs. They're about 150 nucleotides long. Remember, the RNA nucleotides long. And they get together to form a spliceosome, and the spliceosome will actually do the splicing out of the introns and the edit or joining of the exons. Here we have something known as a ribozyme because the RNA is actually acting like an enzyme by cutting and joining. So we have spliceosomes here. The green are the introns, and the red is the exons. I can draw a picture in class if you'd like. Just ask me tomorrow. I'm more than happy to show you how spliceosomes work. So what are the possible functions of introns? Why are they there? Well, they may play a role in gene regulations. For example, the introns might be the siRNA or miRNA that can help regulate protein production. Introns and exons might be different in every cell. So the introns in one cell might be the exons in another cell. So that might be how we can produce different proteins from the 35,000 genes that we have. Now, evolution may be able to change one part of the protein into another part just by keeping the introns there instead of splicing them out. So now we have talked about transcription. So we have here all the DNA, the introns are cut out, the exons are pushed together, we have the 5' cap, we have the poly A tail, and when all that has happened, we have mRNA. So when the introns are cut out, the ends are modified, we have gone from hnRNA to mRNA, and we are ready for what? Well, the mRNA is escorted to the nuclear pores, it enters the cytoplasm, and it's ready for translation. But before that, we need to talk about the code, and we will talk about the code in the next screencast. Have a good night.